Okay, so now I'm going to really go through the techniques. I think that's a big part of the reason why you guys are here, how we actually analyze for biomarkers and analyze for isotopes, and the actual techniques, what you guys can take home and maybe start doing in your research at your home institutes. There are a few different things we're going to go through. The first is obviously collecting the samples, how we get the samples from the field into the lab. Then we have to extract the lipids, because here's our source. The lipids are our source of our N alkanes. Then we're going to do silica gel column chromatography. It's another technique for isolating specific compounds. Then we analyze them with a gas chromatography, flame ionization, and then isotope ratio mass spec, and then we'll talk about pyrolysis and what that means in terms of a reference collection and our data in general. So in terms of selecting samples and collecting samples, we typically take a column approach. Now I don't know whether or not you work with archaeological horizons like this, but our approach is to sample from the different stratigraphic units. So in this instance here from FLK in Olduvai, we usually do a bottom-up approach. We'll expose a fresh surface, clean the surface to make sure we don't have any modern material covering the surface, and what we're excavating, what we're collecting has been preserved over the last however long. These are based off of geological sediments, how they were deposited, the grain size, the rock type, if there are any. And we do, then we can see here, we do a column approach from bottom to top, through the different sediments, and then also horizontally to see if we have any variation between meter by meter. And obviously if we have large variations from this column to this column, then we know we have a problem. Because if this wall here is from the same time frame as we expect, we shouldn't see much variation within a meter because climate just doesn't change at that scale. We also do interval sampling. So in our case, we would take 2 centimeter vertically by about 10 centimeter width and 4 or 5 into the wall. And we usually leave about 4 centimeters in between each sample, and that's just for integrity, because if you go much finer than that, the wall starts collapsing on you, and then you screw up everything. So from the bottom up, we'll take samples at every 4 centimeter intervals through our column through time. So if we can sort of date different units, date for the bottom and date the top, we know we have sampling, chronological sampling throughout. And this really doesn't show up well, but that was at one site. We also did this throughout horizontally throughout the gorge. This is at the BK site in the western part here, and David way in the back actually helped me this summer collect a lot of these samples. He was a great help throughout the gorge. So we did samples horizontally. This is about 2K or so, to see landscape variation. So not only temporal variation, but to see if there were many changes climate-wise or environmental-wise over the landscape itself. And this is not really fun, collecting these samples. Oftentimes you're on your stomach or hands and knees. This is a small dental pick, and it's really a pain trying to collect a lot of these samples when they're almost consolidated, when they're almost rock. So it does take a lot of time and a lot of patience collecting these. So if you have any suggestions or recommendations for collecting samples, I would love to hear them, because this takes way too long to collect a dozen or so samples. Okay, so however you collect your samples, we usually have them as a sediment. Sometimes, if they're more modern, you might have to crush them with a mortar and pestle, just so you can expose more surface area. But if we're interested in carbon and hydrogen isotopes, we really can't have any input from modern, water or modern inputs themselves. So the first thing we always have to do is freeze dry our samples. And the freeze dry, it's a pretty simple technique. It uses sublimation. So it'll take your sample, it'll freeze it, and then it'll change the pressure so it turns that frozen material directly into water vapor, and it'll collect it in this contraption here. It's not destructive, particularly for N-alkanes. I think for some things like starches, it might be a little more destructive, but for N-alkanes and phytoliths, it shouldn't really cause any problem when doing this. You can see how cold it does get, -50, so I know a lot of you are really cold because it's been, what, 3 degrees or so here the last few days. We're in Calgary, we get to -28, -30, so this is actually warm for us this time of year, but we don't quite hit -50, -40 every once in a while. But really, so it really drops the temperature down. So if you do have any water or any other liquid in your sample, it'll freeze it at these temperatures, and then based on these changes in pressure, it'll sublimate it right away. So this is the first step. This is really an easy technique, really minimum sample prep. And what happens is your samples, usually you keep your samples individually in a collection vial or some other means, and the samples themselves go in these flasks. And then you can control whether or not pressure is going to change. You can open and close these valves here, and that allows for the freeze dry, the equipment itself, to extract any of the sublimated gases, any of the water vapor itself. In here, the ice will collect in this chamber here, and then eventually it melts out, and you collect the water afterwards. So once you do that, that's just a great technique. Even if you don't think your sample has any water in it whatsoever, it's always safe to do ahead of time. Then we have to really start worrying about extracting our lipids, our source for our N-alkanes. And we use a piece of equipment called an ACE, an accelerated solvent extractor. Sorry about this. But what we always want to do first, so the ACE is really designed for sediments or even modern plant material. You can extract stuff from modern plants, but you always have to crush your sample first. And the reason for this is to expose surface area. So the more surface area you have exposed, the better your chances are at extracting the lipids themselves. Whether the lipids are collected on sand grains or sediment grains, excuse me, or within the leaf structure itself. So oftentimes you can't see the plant material, but it's in here, it's in this sediment. For really difficult samples, like if you have modern bamboo, for example, I worked on a project, and this will maybe sound silly, but we looked at bamboo as it went through the panda's digestive tract. So we actually had to collect panda poop and analyze the panda poop for lipids. And that's really not, they don't really do a good job digesting their food. So we'd often have to use liquid nitrogen. And just a little bit of liquid nitrogen will help for breaking up your sample. So if you do more modern material, liquid nitrogen, again, it doesn't really affect the biomarkers, the N-alkanes themselves. So it's always great for that. So you just grind up your samples, and usually we like to do, I don't know, between 20 and 30 milliliters of crushed sample. You can even use less for modern material. For the really old stuff, you might have to go up to 60 or more milliliters in terms, but always just make sure to have your enough material depending on age and preservation. The accelerated solvent extractor like this is one that we have at our university. It's a little bit older model, but it still works great. And it's designed to automate compound extraction. So there are many techniques where you can do this manually, and it might take 24 to 48 hours to do. Using one of these instruments, it takes a half hour typically to run one sample. You can load up 12 samples at a time, so within 6 hours you can have 12 samples totally extracted. It can run overnight. You don't have to be there to watch it. It doesn't take much maintenance. What it needs nitrogen gas and the organic solvents that you typically use, and that depends on the sample and what it is you're looking to extract. So in our case, we use these small cells. These are 34 milliliter, we call them ACE cells. We have 66 milliliter and 100 milliliter ones as well. But basically what you do when you take your crushed sample, this piece here screws into one of the caps. These are our septas, our ways to prevent any gas leak or sediment leak from within. And then you fill the tube usually halfway with your sample. If it's older material, you can fill it up a little more. And then you cap it with sand, so clean, oven-baked, natural beach sand, basically. oven bake it at 500 degrees or so, and it should destroy any organics. And this acts as a buffer. You don't want to get any gases in the chamber itself, because if you get any gas leak, it really screws up the sample, and oftentimes it shuts down the machine. If that happens overnight, and no one's there to watch it.
Is it easy for you to summarize what solvents are using for each analysis?
Yeah, of course. So I think, well, I have coming back later. I have a few slides. I use dichloromethane and methanol to extract the N-alkanes, and it's all based on polarity. So if you know you have a really polar compound, you might need to use something a little. Methanol is actually quite polar compound. But really it depends on what it is you're looking for. So for N-alkanes, you can use a 2-to-1 ratio. So you have 2 parts dichloromethane and 1 part methanol, and this is great for extracting N-alkanes or lipids in general. But if you're looking for other compounds, you just change the concentration or the compound itself, and it's really simple.
Have you found, is there an important difference between chloroform and dichloromethane?
I've actually never tried it that way. I don't think so. I know a lot of people who use chloroform, but I learned using dichloromethane and methanol, so I've just stuck to that. And the ratio, some people will do 9-to-1 versus 2-to-1, but that doesn't really seem to show much different in terms of extracting. So how do the ACE actually work? So what is it that's happening? So you fill here's your ACE cell, and you fill it with your sample. And in the back of the machine, there's an oven. And the oven goes up to 150, 200 degrees Celsius. So the cell itself gets loaded in the oven. It's filled with your solvent, and then it's heated to temperature and the pressure change. So it goes up to 1600 psi. And then that pressure helps using the heated solvent plus the pressure is what really extracts the lipids from the sediment matrix itself. And you collect it in a collection vial through time. And it has a lot of different cycles. Usually it runs 3 to 4 to 5 times. So you're making sure to extract every little bit. So the oven turns on, you heat the cell under pressure, really high pressure, and then you start emptying the material. It's flushed through, so you collect the solvent plus the lipids or your desired compounds in your collection vial itself. And again, it's all automated. Once you start the process and you set up the methodology, it does it for you. As long as you don't have any issues with your collection vial itself, sometimes they might not be tight enough. You get some gas leaks, and that shuts it down. But usually you don't have any issues with this on its own. And the resulting output is your solvent and your collected residue itself. Okay, so from there though, so now you're left with a vial that's filled with not only your sample but the solvent itself. So you have to evaporate it out. So typically you use a steady flow of nitrogen gas, and this will dissolve or evaporate your sample over time. You were asking about the solvents. So for the ACE itself, we always use a combination of dichloromethane and methanol. This next step, the silica gel column chromatography, always follows the ACE, because with the ACE, we isolated lipids, but now we need to isolate just the N-alkanes. Or if you wanted to do other compounds, you would select a different solvent. But because the N-alkanes aren't that polar, using a hexane matrix is perfect for what we're trying to do. So really simple setup. You have your, in terms of materials, you have your basic chemical compounds. Always use HPLC grade, so really pure samples, 99% or more, because this really reduces any chance of contamination or introduction of false N-alkanes from other things. Also, we use silica gel, which is a really fine-grained matrix of silica, obviously, and that allows for specific compounds to either pass through the system or they get collected on the silica itself. And then glass wool, oven-baked glass wool to act as a filter. And it's really simple. So the setup itself is simple. So we use 5-inch pipettes, and the pipette is our column. So when we're talking about silica gel column chromatography, the pipette is our column. So we start with the glass wool filter at the bottom, make sure it's blocking so no physical or larger grain particles can actually get through. And then when we fill our sample, or column, excuse me, with the silica gel itself, the glass wool acts as a barrier. So we're only going to extract the lipids or the N-alkanes and any liquid. So in this case, the hexane solvent.
How do you pack the wool?
Yeah, so we have a 5-inch pipette, and then we use 9-inch pipettes. So the 9-inch pipette has, he asked how do we pack the wool at the bottom? So, I don't know if you want to re-ask. But anyway, so the 9-inch pipettes have a much longer tip at the end, so they fit perfectly right in here. So we just gently pack it down with a 9-inch pipette. If you have a metallic spoon or something else that can fit in there, it'll work exactly the same. But with the glass pipettes, we always oven bake them at 500 degrees to remove any organics on them. So we usually oven bake the 5 and 9 mil at the same time. 500. 450 to 500 for 6 to 8 hours should typically get rid of any organic materials. So once that we fill it up with our silica gel, 1 to 200 mesh silica gel, and then we typically saturate the column. So before putting your sample into the column, you saturate it with the solvent you're using. And you flush it through, I don't know, 3 or 4, maybe 5 times, just to make sure that if there's any organic residue that is somehow still in your silica gel, even after oven baking it, you flush it out beforehand. So you would start by, here's where your collection vial would be. You don't have any, this is just an empty bottle. There's no actual residue in there. But you would flush your column. You would collect the first rinse material in a separate vial. And then once your column is completely saturated, you always want to make sure it's saturated throughout the entire run. You don't want to ever let it get dry because that will cause problems for your silica gel. What you then do is you take another pipette, a nice oven clean pipette, and you rinse the vial itself with your solvent. So we typically use hexane. So you would rinse your solvent vial with hexane over and over again to collect the N-alkanes, and then you would add that to the column. And slowly, due to gravity, the N-alkanes will pass through, but the other compounds that you might not be interested in, like the N-alkanols or the acids themselves, they'll get collected in the matrix. Because of their polarity, they'll get attached to the matrix itself. And then at the very bottom, you start collecting the hexane solvent with the N-alkanes. And what's left over in this vial from here, so in here, if we have our total lipids, at the very end of the column itself, you have just the N-alkanes. And that's perfect because that's what we need to analyze both biomarkers and for isotopes itself. Make sense? Somewhat?
Yes.
No? So again, this is just for sample sake. You can do them one at a time and use a pipette holder or whatever, but this was a contraption that was made by a university in China when we did it, and this fits up to 8 or 9 samples at a time, so you can really fly through in a day as many samples as you need to. These are empty, obviously, but really simple, just pieces of plastic and screw bits, and you can just make it whatever you want. Okay, so now that we've separated out our N-alkanes, we have to actually analyze them for abundance and preservation, and we do that with gas chromatography flame ionization detection. So the GC, this is a simple adjuvant model GC, and you can, up here we have our sample injector. And our sample vials fit in here, and it's all automated. It collects the vial and injects it into our oven. So here's our oven where we turn up the temperature to 300, 320 degrees. It passes through the column and the compounds get separated out on their own. The FID is a small portion of it up here where the flame is held, where it's stored, and it actually uses hydrogen gas to light the flame. So you do need a hydrogen generator. This just fills with water. It separates the water from the oxygen from the hydrogen itself, and it uses that to combust the flame. The MS, the mass spec, isn't actually used in this technique. So the one issue with FID is that you can't get the computer to tell you what it is you're looking at, what compounds you actually have. Which the nice thing about a mass spec, it has a data library, and the library will tell you what it is you're actually looking at. For the FID, you have to use a reference standard. So if we know we have an N-alkane reference standard from C C9 to C39 or whatever, we also have to do the exact same technique for analyzing the reference standard with your modern samples. So you have to compare the peak abundance with the time frame and do an actual matchup of, say, something like C25. These are just some typical parameters that we would use for analyzing N-alkanes. It's really a simple setup. It can be used on different techniques. It doesn't have to be a specific GC. It could be anything, really. But we always start out with the temperature. It's something like 60 degrees warm, but not enough where it would actually break apart the samples. And we always ramp up to 320, 325 degrees in the oven, and that is on different time. So as you go through time, you're changing the actual rate of temperature, and that separates out your compounds and gives you an accurate reading on their abundance. And for the FID, again, you need the airflow and the makeup and whatnot. We always use a 60-meter column. You can use a 30-meter column, but you don't get as high resolution towards the end of your sample. So you might only have, say 35, 40 minutes, where you might need 60 minutes to actually run the sample itself. So when the sample is injected, again, it uses nitrogen gas or helium gas as a carrier flow, and this helps the sample pass through the system itself. There are a few different things. So we have a moisture trap and a hydrocarbon trap. Obviously, we don't want this getting into our GC because this will cause contaminants from our original sample. So we always make sure that when you have a setup, there are these traps that collect any modern material that would cause an issue. We always, and this again depends on your sample itself, but we like to fill the insert here with the sample. It's a liquid solvent, so you'd fill that with the hexane, and then this goes into the 2 ml vials. And this is what's collected by the GC itself. The GC, the automated, will pick up the vial and it'll inject it. It'll collect the sample and then inject it into the column. And just looking at the oven, so here's our 60-meter column plus our box oven, temperature related from 60 to 320 degrees. And as it's passing through here, this is when we start separating out the compounds. So early on, we have here's maybe our different compounds, N-alkanes or C27, 29 or whatever, and they're all together. They're injected at the same time. But as you change the temperature and you change the gas flow, they start slowly getting separated out. And then towards the end, you really have separation based on molecular weight. So heavier items take a little bit more energy to break apart and separate out, whereas lighter stuff, like hexane, for example, is a low molecular formula. It would be shown early on. And then our resulting output is a simple chromatogram from time and then compound abundance. So here's an example of a sample. This might, actually I don't remember where this sample is from actually. But anyway, so what you would happen is you would have, I showed this earlier, but then you really have your peaks identified in terms of relative abundance based on time. And if we're interested in the N-alkanes and the hydrocarbons themselves for isotopes, these three examples are perfect for analyzing. They're well preserved. There's quite a lot of them. We know that their biosynthesis is from terrestrial plants. From there, we then have to look at the actual isotope compositions, and this is really what's showing us what was going on climatically. And this is a little bit more complex. The GCFID is in terms of cost, it's much cheaper to have an FID at a university or an institute than the isotope mass spec, because the isotope mass spec takes a lot. You need a proper technician to run the equipment, and it's more than just one contained unit. You have a lot, multiple different instruments being used at the same time. And typically, this can cost money if you have to send your samples away to specific places. It does, the cost can add up, especially if you have 30, 40, 50 samples to run. But again, it's a very similar idea. You set your temperature parameters. It uses a few different gases. In this case, we're looking at hydrogen and carbon. And carbon's been used for the last, 40, 50 years for isotope analysis, but hydrogen on its own has only recently been used, maybe within the last 20 years. And it was always a little bit more difficult to understand hydrogen's abundance in your samples because you need a lot more of the sample itself. Something like carbon, you can dissolve 50 nanograms of carbon to get your isotope value, where for hydrogen you might need 250, 300 nanograms to get the same results. So our output for isotopes is very similar to what you would see with the GCFID. So again, we have our compounds, so here's 27, 29, and 31. But instead of identifying the relative abundance of the hydrocarbons themselves, now we're looking at the ratio between hydrogen and helium. So the machine will really select out the individual compounds themselves, but it'll give you an understanding of that ratio, that delta D between the heavy and lighter isotope.